My name is Catherine Amoateng. I am from the Department of Chemistry and Biochemistry. For the next few minutes, I'll be talking to you about a design undergraduate experiment that uses the color of precipitates to quantify known trace compounds in solution. The rationale behind this lab is seen in the limited use of precipitation chemistry in our teaching labs, as well as the current enhanced imaging sensors in digital microscopes. The design experiment therefore takes advantage of color and quality of digital microscopes. The design experiment is a one-step precipitation reaction after which a 1 microliter aliquot of the solution mixture is taken and dried. To isolate the compound of interest, the dried precipitate is washed with DI water and imaged again. The color intensity of the dried image is then processed using ImageJ open source software. The learning goal is to have students quantify a trace amount in an aqueous solution using the color intensity and image of the precipitated ionic compound. Using these simple experimental steps, students in a lab are asked to quantify zinc gluconate, which is the main ingredient in a zinc tablet, through these 4 steps. 1. They prepare an ionic zinc calibration curve using the yellow color intensity of microscopic images of ionic zinc standard solutions precipitated as ionic ferrocyanide, as seen in the equation below. Then they extract and precipitate ionic zinc from zinc tablets, which is then imaged and processed for color intensity. The generated calibration curve of the standard ionic solution is then used to quantify the ionic zinc concentration in the zinc tablet. Finally, students calculate zinc gluconate content using the ionic zinc concentration obtained from the calibration curve, as well as the weights and dilutions from the ionic zinc extraction steps. The implementation of this design experiment could not be done in spring this year, 2020, due to the coronavirus pandemic. The first 2 columns here could not be done when classes went remote. In its place, ionic zinc images of standard solutions and samples were given to students to process. In addition to the images given to students to process, a set of selected pre-lab questions were given to students to assess students' prior knowledge in predicting precipitation formation in solutions containing trace amounts of cations and anions. For each ionic zinc standard solution, 2 images representing a pre-rinse image and a post-rinse image are given to students. The ionic zinc precipitate is confined inside a solution aliquot. When this dries up, the pre-rinse image is taken before washing to isolate the compound of interest seen in the post-rinse image. The pre-rinse image is used to identify the region of interest of the post-rinse image. As you can see, the outline of the post-rinse image is very faint and very difficult to identify. And so the pre-rinse image outline helps students to identify the outline for the post-rinse image. The sum of the yellow color intensity is then processed using the post-rinse image. When the post-rinse image is processed through the ImageJ software, a color histogram output is given showing a range of colors. The yellow color represents ionic zinc, and to obtain only the summation of this yellow coloration, numerical numbers from 50 to 60 degrees, representing all shades of yellow on a color model is used. A summation of all yellow-colored pixels is obtained, and this correlates to the concentration of ionic zinc. With the learning goal of quantifying trace ionic compounds in aqueous solution using color intensity of microscopic digital images, all 10 groups of students working in groups of 4, assessed by the rubric, met the learning goal. 3 groups of students, however, exceeded the learning goal. Here is a group report that exceeded the learning goal. The students have their calibration curve plotted, and they are showing their R-squared value and the equation of the line that was used to calculate the ionic zinc content of the sample. The students, however, discuss the accuracy and precision of the data sets that they have at hand. I will give you a few seconds to read the conclusion of this discussion. This is highlighted in yellow. In conclusion, the remote form of the design experiment was successfully implemented in an analytical undergraduate course. Evidence provided shows students can demonstrate competence of the learning goal in processing digital microscopic images of precipitates and quantify a metal in aqueous solution. There is little evidence, however, to show students' comprehension of the microscale nature of the analysis. Modifications will be made to the pre-lab and the post-lab tasks to help students comprehend the microscale nature of the lab. We thank the students and instructor of Chem 375 Analytical Chemistry for their participation and help in implementing this experimental activity. We are also grateful to the Yezierski and Brett's research groups for their feedback on earlier versions of the laboratory activity. We also thank Miami University for funding this research. Thank you for watching.